Hey there guys, it's Justin, your biology laboratory technician. I'm in your genetics laboratory and I'm going to do a demonstration on how to identify genetically modified foods using PCR. And I'm following the protocol from this kit by a company called Edvotech. There's their logo there on the bottom. And I'm using EdvoKit number 962. So this is something that if you wanted to follow along with the instructions as I'm doing them, you can look them up online and kind of follow what I'm doing here. So this is just a demonstration on what you guys would do in the lab, and hopefully what we're going to get out of this is to be able to determine the difference between common food items as far as if they contain genetically modified products such as corn, soybeans, those are some popular ones, or not. There's a lot of items obviously still out there that have not been genetically modified, so the purpose of this lab is to get some food samples from home and see if they contain certain indicators that they have been genetically modified or not. So I'm just going to test one food item. So I have some corn muffin mix here, and positive that these are going to have most corn today, if not all, has been genetically modified at some point to help in agricultural, for agricultural purposes rather, to withstand weather, to withstand natural and unnatural forces out there, to have farmers have a better yield in their corn, and also possibly to have more nutrients and just have more attractive looking crops. And so most corn and corn products, such as this corn muffin mix, are going to contain something that we could find in there that will indicate that it is a GMO. In this case, we're isolating the DNA from food items, and then we're going to run polymerase chain reaction in our thermocycler, and what that's going to do is using our specific primers that comes with this kit, it's going to be able to attach to certain regions of the genome of the DNA that we isolate and amplify the region that we're interested in, that will tell us whether or not it is in fact a GMO. So I'm just going to walk you through the steps here. So yeah, I'm using the corn muffin mix for a probable GMO source, and then a certified non-GMO source in this case came with the kit, and these are pine nuts, and we're going to use this as a negative control in our experiment. So we want to see what it looks like to not have GMOs as a PCR product. Okay, so over here I have all the stuff I'm going to need. I have a tube heater right here set to 56 Celsius. I have a microcentrifuge here. I have a laptop cooler with some reagents that we're going to need to keep cold during this. I have my pipette tips, a vortexer, and my pipetters over here. And then also I have a DNA extraction buffer that came with the kit and sodium chloride solution. In my tube rack here I have some micro pestles that we're going to use to grind up our food products in the tube. I also have isopropanol and 70% ethanol. I'm going to set these aside now and then show you. So I have my negative controlled pine nuts here that I've ground up on some clean paper. And then I have my cornbread mix right here, which I've also ground up. Okay, so our first step then is going to get the food samples that we're going to be testing into our initial tube. So that's what we're going to do first. So I'm going to first put my pine nut food sample in the tube up to the 0.1 milliliter mark here, because we don't need too much. So let me zoom in here. Okay, so I have some on the end of my pestle here. That should be plenty. I'm going to set this aside. I'm going to designate one of these micropestles for each of our specimens because we don't want to cross-contaminate. So I'm just going to set this tube down and then gently get some of the product in the tube. And I'm just scraping some off the end of here. See how much we have. I'm just going to make sure it's all pushed down to the bottom of the tube. And it's going right to my 0.1 milliliter mark there. So we are good. So that's 100 microliters of dry food product there in my pine nuts labeled tube. Set those things aside and then do the same thing for the corn mix. Have my clean micropestle here and gently get some in my tube. That's looking good. Get it all down in there. I could probably use a little more. Just a tiny bit more here. There we go. Alright, so I have my cornbread mix up to the 0.1 milliliter mark in my microcentrifuge tube there. Okay. So next, we want to add a DNA extraction buffer. So this is just a buffer that's going to promote the extraction of DNA in our organisms here, which are corn and pine nuts. Also, what is in this extraction buffer, which I added before I started, is what is called proteinase K, and that is just a reagent that's going to help degrade any proteins that are surrounding the DNA that we're interested in. So in these samples, you know, you're going to have a lot of other cellular debris and proteins and unwanted stuff that we are not concerned about right now, and we just want to get to that crude genomic DNA, and that's what this buffer and the proteinase K helps promote. So to each of the samples, I'm going to add 400 microliters of the DNA extraction buffer. So I'm going to open my tubes up. And I'm going to grab my pipetter and set it to 400 microliters. Lock it in place. Give it a check, make sure it seems right. That seems good. Actually need the bigger tips for this one. Okay. So 400 microliters of the DNA extraction buffer into each of our samples. Okay, so you'll notice that I inserted the liquid from above the sample, so I wasn't touching the inside of the tube, so I'm not cross-contaminating these. Okay, so I'm going to close the lids on this, dispose of my tip. Okay. All right, now we are going to grind this food sample really well with the same micro pestle that we used to put the sample in our tube. Okay, so just grind this for about 30 seconds and, just really apply a lot of pressure. Try not to overflow, don't get too crazy and spill it all over the place, but just kind of press down and spin. Until it's a nice homogeneous mixture. And this is just really helping distribute that extraction buffer to come in contact with every little surface of our sample to help promote the extraction of the DNA in there. Okay, that seems good. Do the same for my other sample. Using a different micro pestle, obviously, because we don't want to cross-contaminate. Just grind it up really good. When you're done grinding, it should all look the same in there. You shouldn't see any big floating chunks or anything like that. It should all look nice and homogeneous. Okay, close the lid. Okay, so now I'm going to use my vortex mixer over here and vortex for about 30 seconds just to get a really good mix. Okay, give it a couple flicks, make sure it's nice and mixed up. That looks good. Okay, now my pine nut sample, 30 seconds on the vortexer. Turn this up a little. There we go. Okay, That looks good. Give it a couple flicks, make sure there's no big chunks floating around. Looks great. Okay, now we're going to incubate these samples in our tube heater right here that's set to 56 Celsius. So you just put them right in the tube rack, and we'll let those incubate for 15 minutes at 56 Celsius before we add our sodium chloride solution, which is the next step. Okay, it's been 15 minutes. I'm going to remove my tubes from the 56 Celsius tube heater over here. And we can see how the heavier sediment of the food stuff has sunk to the bottom, just based on mass alone, and you have this liquid layer on top. So we'll observe that. We'll put these back in our tube holder here. Okay, and now we're going to add 300 microliters of sodium chloride solution and then mix that. So we're going to set our pipette to 300. Okay. Get a fresh tip. Open up our tube caps. And disperse 300 microliters into each. Without touching the inside of the tube or making sure it doesn't splash back up, we will insert 300 microliters of the sodium chloride solution, which is going to neutralize everything in there. And it's just going to promote the separation and the forming of a DNA pellet after we add the alcohol, which will actually precipitate the DNA. Okay, so we have our sodium chloride solution. I'm going to vortex each sample for about 30 seconds just to give it a good mix and make sure all the surfaces are covered. Zoom out a little. And give it a couple flicks for good measure. And my other sample. Okay, nice and mixed. Okay, now we're going to use our microcentrifuge at full speed. So full speed on our mini spin over here is 13.4 RPMs. And since we're only using 2 samples, we won't need a balancing tube in there. So I'll take that out and put these samples directly across from each other with the hinge pointing away from the center of the centrifuge. Make sure both of them are lined up. Put the lid on and let this spin for 5 minutes. And I've set the timer on here for 5 minutes already, so all I have to do is hit start. Okay, our centrifuge is slowing down now. We spun it for 5 minutes at full speed. So now what we want to do is carefully remove these from the centrifuge, and we're going to look for this pellet at the bottom. So it's a very large pellet right now because pretty much all of the heavy stuff has been centrifuged down to the bottom of the tube, and what we're interested in is the supernatant layer on top. So I'm going to be careful not to shake these around too much and dislodge that pellet. Let's look at our other sample here. Okay, we do have some floaty stuff on top, so we're going to try to ignore that and just remove the liquid supernatant. And I have pre-labeled new tubes that we're going to insert our supernatant into. So I'm going to do my corn sample first. Open up my new tube. Let me fix the view here. Okay, so I'm going to be removing about, if we look at the graduations on here, I'm going to estimate about 400 to 500 microliters of the supernatant to put in the new tube. So I'm going to go ahead and set my pipetter to 400 microliters and get a new tip. And gently tip my tube here with the angle of the supernatant facing me, and I'm plunging my plunger down on my pipetter first before I go in, because I'm going to remove liquid. I'm going to avoid getting too many chunks in here. So if you look at the liquid in my pipette tip there, it's mostly clearish color, so that's good. I don't want too many larger debris in there, so I can get a little more out of here, I think. Just a little bit. Okay. Now we're done with this tube. Set it aside. Okay, now my pine nut sample. Open up the new tube. Examine this and see where I'm going to pipette from. Get a new tip. And remove the supernatant from this tube. Okay. Probably get a little more out of there. If you get a couple chunks in there, it's okay, because there's going to be some more centrifugation steps left. You just don't want to get the stuff that has sunk to the bottom there. Okay, so now we're going to see how much volume of supernatant that we removed, and we're going to add that volume amount of isopropanol to precipitate the DNA. So it looks like I have here about 500 microliters for the corn and about the same, 500 microliters for the pine nuts. So I'm going to put 500 microliters of isopropanol into each of those tubes. And the isopropanol I'm using is at room temperature, so it doesn't have to be cold at this point. So get a new tip. Get 500 microliters of isopropanol. Put it in my corn sample. And 500 microliters for my pine nut supernatant. Okay, so now we're just going to let this sit. Close the caps, but you can see how it's turning a cloudy color. So it was a little cloudy to begin with because there was still some debris from the food in there, but now it's really starting to get cloudy, and that is potentially the DNA precipitating out of the supernatant. Okay, so I'm going to mix this up so that isopropanol comes into contact with all of the supernatant, and I'm just going to vortex over here. And it should look like that. It's like a cloudy looking solution. Okay, we're going to let that incubate at room temperature for 5 minutes. Okay, we'll let that sit for 5 minutes at room temp, and we'll move on to the next step. Okay, so we've let our precipitated DNA sit at room temp for 5 minutes, and if we examine the appearance of these guys, we can see a couple layers that have formed in here and a lot of floaties in there, so hopefully we have a lot of DNA precipitated. So what we want to do is concentrate it all to the bottom of the tube by centrifuging again at full speed for 5 minutes. So we'll pop them in our microcentrifuge here. Make sure they're evenly spaced with the hinges facing out. And the narrow part of the tube facing in towards the center. Put the lid on and let it spin. Okay, as our centrifuge is finishing this round of full speed centrifugation to form the DNA pellet at the bottom of the tube, we're just going to examine these and look for the pellet. Alright, so carefully move it, remove it from the centrifuge, and look on the bottom for a pellet. You may not be able to see it here, but there is a pellet right there on the bottom, so be careful not to jostle that too much. That was the pine nut sample, and this is our corn sample. and I can see a tiny little pellet at the bottom of this one as well. Tiny is good in this sense because, as we know, DNA is molecular size and it is very small. But if we can actually visualize a pellet in this case, that's potentially a good concentration of DNA. And the more concentrated DNA we have to use as a template in PCR, the better. Okay. Alright, so next, let me fix the camera back there, zoom in a little bit, and so we have formed our DNA pellet, and now we want to remove and get rid of the supernatant. So before we were keeping the supernatant, now that we've precipitated the DNA in our solution and have pushed it all to the bottom of the tube by centrifugation, now we want to get rid of all the supernatant since our DNA is now potentially on the bottom. So to do this, we're just going to decant off the supernatant here as well as we can without disturbing the pellet on the bottom. So I'm going to use a pipetter and a new tip. And I have it set to 500 microliters because I don't want to be too forceful in removing the supernatant here. So I set it a little less volume than I have in the tube, just to be careful. I'm just going to squirt that in my waste beaker over there out of view. And if you see any chunks, like cellular debris and other possible stuff that we're not interested, go ahead and try to remove those like I have in this pine nut sample here. Anything that's not at the bottom of the tube we want to get rid of. Okay, that one's pretty good. I'm going to get a new tip because I don't want to cross-contaminate. And I'm going to remove the supernatant from our corn sample. I'm going to keep an eye on my pellet so I don't accidentally bump into it. And then decant all of that. Then I'm going to visually inspect this again just to make sure I still see the pellet that I formed. And everything else looks good. I do have a little film here sticking to the side, which is just some unwanted debris, so I'm going to go ahead and get another tip and physically remove that from the tube because we don't want that. I'm just kind of scraping it with a sterile pipette tip there. Okay, looks pretty good. Alright, so now we have concentrated DNA on the bottom of the tubes, and we want to kind of rinse this off and degrade any proteinaceous or cellular material that's still in there and wash it with 70% ethanol. So that's what I have here in this tube, and I'm going to add 500 microliters to each tube, but I do not want to dislodge that pellet on the bottom, so I'm going to add it really slowly to the side. So 500 microliters, and I'm just going to pick up my tube, tilt it, and insert it and just slowly let it run down the side of the tube. Because if I squirt too hard, you know, it'll wash that pellet up from the bottom, and we don't want that. We want to keep it down there for now. Okay, repeat for the other sample. 500 microliters of 70% ethanol for washing our DNA pellet. Okay, so now we want to centrifuge it again. So our pellet's still on the bottom, but now we want to completely wash it and let it come, all the ethanol come in contact with everything on the bottom of the tube. So we're going to centrifuge it again at full speed, but this time for only 2 minutes. So I'm just going to insert my tubes over here. Again, with the hinges pointing away from the center of the centrifuge. Make sure they're balanced, evenly spaced. I'm going to change the time on this from 5 minutes to 2 minutes and let that spin. Okay, so we spun our washed DNA pellet. We did a final spin at full speed for 2 minutes. So let's examine these. And I can see a really tiny dot of solid material down there, which is what we're interested in. That is our concentrated DNA pellet. So again, be careful not to jostle it too much and disrupt that pellet. And the same here, the pine nut has a lot of material on that. I'm assuming not all of that is DNA, so hopefully we washed it enough and got rid of a lot of the cellular debris and protein, so when PCR time comes, the primers can easily find the genomic DNA on here. But irregardless, we're going to now decant all of that ethanol off of our samples and then let the DNA pellet air dry until it's completely dry, and then we will resuspend it in a DNA universal buffer, which is this stuff. Universal DNA buffer is just a nuclease-free reagent, or just really an inert solution that will protect our DNA, and it ensures us that there's no nucleases in it, or like DNase or nuclease, anything that's going to degrade our super sensitive DNA product. So that's all this DNA buffer is. In place of this, you can also use a nuclease-free water, which is often used in the research laboratory. While that thaws out a little bit, I'm going to carefully decant all the ethanol, just as we did before with the isopropanol, but this time, the same thing. We're going to just stay away from our DNA pellet and try to get as much as this out of the tube as possible without disturbing that pellet. So we'll get a new sterile tip. Put Put my plunger down, go in, keep my eye on the pellet, and remove this ethanol. My pipetter is set to 500 microliters. And I'm just going to completely remove all the liquid from that tube. And now here you can see, maybe, see that little tiny white dot at the bottom of the tube? That is our DNA. So I'm just going to leave this tube open and let that ethanol evaporate and just leave the DNA pellet behind. Alright, onto our pine nut sample. Okay. Okay. So I'm going to let the ethanol evaporate off of our DNA, let them sit out, and then what I'm going to do is add 50 microliters of this universal DNA buffer, and then it's safe to freeze these samples. So the next video, we're going to use these as DNA templates and do a PCR reaction, and I will show you how that's done as well on the next video. Thanks, guys.